So yeah, it's my great pleasure to welcome you all today, to welcome David Nathanson here for our Cancer Neuroscience seminar series. And so David Nathanson is an associate professor of molecular and medical pharmacology at UCLA. He received his PhD under Dr. Paul Mischel, where he discovered that extrachromosomal DNA can be dynamically eliminated to promote drug resistance in cancer. He conducted afterwards his postdoctoral work in tumor metabolism, drug development, and molecular imaging with Dr. Caius Radu, leading to the discovery of new clinical drug candidates to target DNA metabolism in cancer. And his independent lab investigates the dynamic interplay between specific molecular alterations and critical functional processes in malignant brain tumors, with the goal of developing new drugs and strategies to exploit these functional vulnerabilities for improved patient outcomes. I'm very happy that he's here today with us to talk about therapeutic resistance and brain cancer neuroscience. And with this, the stage is yours, and happy to have you.
Great, thank you. Is it still showing the regular, not the notes section?
Yes, the regular.
Okay,
the regular.
Awesome. Well, thank you, Varun and Frank and the rest of the organizers of this wonderful meeting. I appreciate the invitation, and of course, all of you on Zoom who are attending. We're talking about something that we kind of stumbled onto. You know, we're looking at how cancer cells, and specifically glioblastoma cells, respond to drug perturbations and how that impacts cell fate. And it kind of led us to, looking into how neuroscience might influence that. So I'm going to talk a little bit about that today, some of the early unpublished data that we've acquired. Let's see. Let's go forward. Here are my disclosures. So of course, the goal for any cancer therapy, whether it's targeted therapies, immunotherapy, chemotherapy, or radiotherapy, is to eliminate the tumor and eradicate all the tumor cells that exist, and this can only be achieved through promoting cancer cell death. Here's an example in clinical where you see this metastatic, these metastatic lesions in the lung from a testicular cancer patient prior to chemotherapy, and then a few months post-chemotherapy, all these lesions have disappeared, and this inevitably led to this patient entering complete remission for over 9 years. So this is an example of a clear example where the cancer therapy is initiating tumor cell death. In glioblastoma, unfortunately, we don't typically see responses like this following a complete surgical resection and the initiation of standard of care consisting of radiotherapy and temozolomide. We may see this kind of transient growth inhibitory effects or stable disease within the tumor, but very quickly and inevitably, this tumor does recur, and this ultimately leads to this dismal survival that we're all familiar with of just over a year for patients with IDH wild-type glioblastoma. We see similar kind of transient effects in our preclinical models, where we take patient tumors and graft them into the brains of mice, create these orthotopic xenografts, and we'll treat them with components of the standard of care, including temozolomide and an MGMT methylated xenograft, and we're able to get a significant survival benefit with both temozolomide as well as radiotherapy in these two unique models. But you can see that the survival is not durable. We're not seeing any cures here within our preclinical model systems using drug doses that are within clinically relevant exposures. And this may be due to the fact that, like what we see clinically, we're really not seeing massive tumor shrinkage. We're seeing primarily what we would call cytostatic effects. So here is the vehicle and the temozolomide treatment. You can see that these growth curves are separating in a significant way, but relative to the starting point, we're unable to actually shrink the tumor. So this is what we're calling a cytostatic response, in contrast to a cytotoxic response where the tumor is actually shrinking. And again, similarly, we see a cytostatic effect in these models with radiation treatment as well. So with these therapies that we're all familiar with that essentially target the DNA and add DNA insults, we're not seeing considerable cell death. What about in the context of targeted agents? Well, one oncogene in particular that's a notable driver of glioblastoma is known as the epidermal growth factor receptor, or EGFR. which is altered in the majority of glioblastoma patients. And when hyperactivated, it signals downstream through canonical pathways such as PI3 kinase, mTOR, as well as RAS-MAP kinase pathway to support glioma growth and survival. So this is a bona fide oncogene and a clear driver of glioblastoma. And so working together with Tim Clousey, we recently discovered a novel brain-penetrant EGFR inhibitor that we tested in a big panel of patient-derived xenografts, over 30. And this is just looking at a waterfall plot of the survival benefit with this therapy. And we're able to see a quite dramatic increase in median survival, around 75% on average, between of the models that responded, and a really encouraging sign specifically within the EGFR altered xenografts of over a 90% increase in survival relative to vehicle. So a potentially attractive targeted therapy, but like what we see with the TMZ and the radiation, the DNA damaging therapies, we're not seeing that these tumors shrink relative to their starting size. You can see this model that had a 30% increase in overall survival. Again, this cytostatic effect, so we're inhibiting growth, but we're not dipping below the starting point. Same thing, even with a 140% increase in survival, it's largely cytostatic. And so the thought is that if we're unable to actually kill these tumor cells, we can't expect to get really durable responses with these therapies in the clinical setting. And so that goes to the question is, what is responsible for precluding cell death when we come in with these therapeutic insults against our malignant cancer cells? And it really, in many cases, begins with this pathway known as the intrinsic or mitochondrial dependent apoptotic pathway, which is frequently utilized by cancer cells to essentially initiate apoptosis after a therapeutic insult. And so the way that this pathway works is after a specific therapeutic perturbation, this can promote the expression of these pro-apoptotic family members, namely BIM, BID, and PUMA. They go and bind to these effector proteins back and backs, stimulate their oligomerization at the mitochondria, and promote the release of cytochrome C and the initiation of the apoptotic cascade, which inevitably leads to apoptosis. However, this pro-apoptotic push is antagonized by these anti-apoptotic proteins, namely MCL1, BCL-XL, and BCL2, which combine and inactivate these pro-apoptotic proteins, and essentially shifting this apoptotic balance to a stronger anti-apoptotic phenotype and preventing cell kill despite the presence of strong chemotherapeutic perturbations. And so the question that we had, because it really just hasn't been well explored in glioblastoma, is does the intrinsic apoptotic machinery contribute to GBM drug resistance? And so to begin to actually answer this question, we kind of need to understand a little bit more of the details surrounding how the interactions between these anti and pro-apoptotic proteins occur. And there's quite a bit of differences in their protein structure and sequence between these anti-apoptotic proteins and the pro-apoptotic proteins. But there is one shared feature, and this is this conserved BH3 domain. And it is through this conserved BH3 domain that these anti-apoptotic proteins and the pro-apoptotic proteins interact on this one-to-one basis. And you can kind of see an example of this with this anti-apoptotic protein BCL-XL interacting with a peptide derived from this pro-apoptotic protein BIM. And so by understanding that these two things are interacting in this way, these dynamics, we could start to make predictions, and this has been tested experimentally, about how this might influence their essentially ability to undergo apoptosis. And so you can imagine that when a cancer cell has much of its anti-apoptotic proteins occupied by these pro-apoptotic proteins, you are essentially closer to apoptosis. You have to overcome these anti-apoptotic blocks. And so if you have more of these pro-apoptotic proteins bound to these anti-apoptotic proteins, you are essentially closer to that goal, and what we designate those cells as being highly primed. Conversely, in cells that have only a limited number of these anti-apoptotic proteins occupied by these pro-apoptotic proteins, you have a lot more of these spaces that need to be occupied by these pro-apoptotic proteins to essentially overcome this anti-apoptotic block. and what we call these cells as being low primed, or they're further away from apoptosis. And so in theory, the way that this would then work, that if these high prime cells, that if they are now to receive a therapeutic insult, that this would then you get these pro-apoptotic push that would overcome this anti-apoptotic block, shift this to this induction of apoptosis, and this would initiate the cell death cascade. Conversely, despite a potentially strong therapeutic insult, the cells that are low prime will only modestly fill up the remaining anti-apoptotic blocks, and you're still further away from apoptosis than you want to be, and these cancer cells end up surviving. In a more kind of simplified way of seeing this, you can imagine cells on a cliff, where a high prime cell is on the edge of the cliff, and it takes a relatively weak perturbation to push these cells over the cliff to achieve cell death. In contrast, when you have this unprimed cell, if you come in with that same perturbation, the cell just might move along the edge of the cliff a little bit, but it's insufficient to actually push it over, and these cells will survive. So these cells have a much lower apoptotic threshold that needs to be overcome to achieve apoptosis. So the whole idea then is, how can we potentially measure whether or not a cell is in this state or this state? And so working with Anthony Lattai at Harvard, who developed this really innovative assay called B-tree profiling, it can actually quantitatively measure how primed a specific cell is for apoptosis. And the way that this works is you take live cells from whatever origin, you permeabilize them, and you incubate them with increasing concentrations of this pro-apoptotic protein, a peptide version of this pro-apoptotic protein BIM. And essentially all you're testing is determining the level of occupancy of these cells with the pro and anti-apoptotic blocks. So here's an example where these cells are already fairly well occupied, and so it takes less BIM to occupy the remaining anti-apoptotic blocks, where here this requires more BIM peptide to occupy the remaining apoptotic blocks. And then as a consequence of that, what we can then measure after titrating in the BIM is we can measure the release of cytochrome C from the mitochondria by flow cytometry, and essentially assess how prime these cells are for death. And so here's a real example where here is a high prime cell. You can see once you start with a very low BIM concentration level, you start to bring in BIM, and you can start to see the release of cytochrome C at very low concentrations, and then you've seen essentially maximum depolarization around 1 micromolar. In contrast, the lowly prime cell, you require a lot more of that BIM peptide to promote the release of cytochrome C. So this one right here would be designated as low primed, and this cell right here that we have would be designated high primed. And Anthony Lattai has shown some pretty intriguing data showing the relationship. Again, this is pre-treatment priming. These are untreated cells, and then having the predictability of response to various chemotherapeutic agents. So this is using AML size and AML models. And they're using this BIM peptide. You can see these various degrees of apoptotic or priming. And then you can see very nice correlations between essentially the priming value and the ability of these various chemotherapeutics in inducing apoptosis. He's actually been able to identify relationships clinically where patients that were measured pre-treatment for their level of priming in both myeloma and ovarian cancer patients, we see that those patients that are high primed seem to associate with a greater response to therapy than those are low primed, and we see a similar effect with ovarian cancer patients. So it seems that this assay seems to do pretty well in predicting preclinical responses and potentially having important clinical implications as well. And so with this knowledge in mind, one of the questions that we had was, is the low prime state essentially associated with a poor response to therapeutics in glioblastoma? And so to begin to explore this question, we've started to embark on taking up both a molecular and functional characterization of the intrinsic apoptotic machinery across patient samples freshly resected from patient tumors. And so from 29 glioma patients, this includes both high grades and low grades. We'll get these bulk tumor resections from neurosurgery. We will purify the tumor cells through depletion of red blood cells, myelin, and infiltrating immune cells. Again, here we're getting a fairly pure tumor population. We get around 95%. And from that, we perform B-short profiling on these live tumor cells. We'll do RNA sequencing and whole exome sequencing to really try to characterize both the functional features and the molecular features of these tumors. And here's an example of what we've kind of had so far. So these are, again, various degrees of IDH status and grades, MGMT methylation, and also molecular features. And what we can initially see from looking at these patient tumors is there's also a varying degree of apoptotic priming, where here's, for example, a more lowly primed GBM relative to a more highly primed GBM. And so, you know, we're a little bit underpowered to start asking specific questions relating molecular features to priming at this moment. But what we certainly have learned is that these gliomas exist in a very large range of apoptotic potential or prime states, as you can see here, looking at these curves, and then summarized here by just looking at the area under the curve of essentially prime state. And here you can see tumors that are lowly primed, and then tumors that you would designate as being highly primed. And just to start investigating some of these questions into what this diversity may mean, one kind of obvious one is because we have a good mix of high grade and low grade gliomas, we see that the high grade gliomas are less primed or less potential to undergo apoptosis than the low grade gliomas. And this seems to be associated with, we of course all know, is that these high grade gliomas have certainly a worse outcome than these low grade gliomas. It certainly doesn't say anything about a causal role, but there seems to be this association between at least apoptotic potential and clinical outcome. We've similarly looked at tumor recurrences, which, as we know, the longer a patient is under treatment and these tumors persist, that the more likely that these patients are going to succumb to the disease. And so here we actually have matched patients looking at recurrences. And you can see from recurrence 1 to recurrence 2, we're essentially seeing that these tumors are becoming less primed, indicating that their ability to undergo apoptosis has diminished over time of being in that brain. And so these were kind of some intriguing findings that what we were seeing clinically and that we're exploring further. But you know, some of the other questions that we've started to explore, which is more the focus on today's talk, is we really wanted to understand the models that we work with every day and try to make translational discoveries with. Do they also show similar intrinsic apoptotic potential or resistance to apoptosis as the patient tumors? And so as we were getting from these purified tumors, concurrently we created these patient-derived cell culture models, which are commonly known as gliomosphere cultures, as well as orthotopic xenografts. And then we've also done some subcutaneous xenografts. And from these various kind of derivatives of the patient, we then perform B-tree profiling, RNA sequencing, and whole exome sequencing as well to really start to characterize our models and see how well they compare to our patient systems to understand the translatability. And one of the things that kind of stood out with us in a very obvious way was when we started to compare our patient tumors to our cell culture models. And essentially what we found, well, again, we're looking at 29 patient samples versus 26 gliomosphere cell cultures. We find that the gliomosphere cell cultures, this is the cytochrome C, is the priming of them, is the gliomosphere cultures are more primed for apoptosis than our patient samples. And so that was kind of intriguing to us. You might think that maybe the gliomospheres may be more primed because they're replicating so quickly, so maybe there may be a little bit under more stress, but we saw that it actually was the opposite. You know, there's so many differences between patient and gliomosphere cultures that we certainly couldn't make any conclusions just from this. So what we did is we took the cells and put them back into the brains of mice, back into trying to match what their native environment was. And quite surprisingly to us, what we found is once you put these cells back into the brains of mice, we are able to dramatically reduce the apoptotic potential, as you can see here with these curves. So the cell culture, this one cell culture model, this is its prime state. So it takes less BIM to promote the release of cytochrome C. And then now, once you go into the brains of mice, you've now reduced its apoptotic priming. And we see that in another GBM model as well. So what this is telling us is now, whether you come from the patient brain or the mouse brain, relative to cell culture, which is closer to the edge of the cliff, once you go into that brain environment, we seem to reduce the apoptotic potential of these glioma cells from undergoing apoptosis. But clearly, there's still a lot of differences between a cell culture environment and both a patient and a mouse in vivo environment. So we wanted to try something somewhat intermediate and see if the brain really created some sort of this unique phenotype. And so what we did is we injected one of our models subcutaneously in the flank, and we took these tumor cells out and again looked at apoptotic potential priming. And what we found was, in contrast to the intracranial models, once you put these cells subcutaneously, we're able to prime them again. So these guys are now closer to the cliff than where you went into the brain. And so now this is really kind of starting to suggest to us that the location of these cells in the in vivo environment really can dictate apoptotic potential, because now, relatively speaking, everything is fairly well matched between the flank and the brain, and the things that are really distinct between those two in the mouse setting is just the environment that these tumor cells are placed in. And so this led us to hypothesize that potentially these tumors in the flank may be more sensitized to these tumors in the brain to a therapeutic induced cell death. And so we began to test that. Again, we took an EGFR mutant patient tumor, we implanted it both in the brain as well as in the flank, and we gave these mice EGFR inhibitors after they've established rapid tumor growth. And just to make sure there weren't any caveats associated with exposure, we find that both, because this is a fully brain penetrant drug, we find that the accumulation in the brain and the subcutaneous tumor of this drug is identical, it's not significantly different. However, we see drastic differences in responses to these tumors that are just placed in different environments. And so here's an example, again, another cytostatic tumor response once it's in the brain of this low primed. But once we put this same tumor subcutaneously, and this is on a log scale, this tumor essentially shrinks to nothing. And so we're able to now convert from a cytostatic response to a cytotoxic response, just moving these tumor cells between the brain environment into the flank environment. And so that really started telling us that maybe these environments really can have a significant impact on whether or not a tumor lives or dies in response to a therapeutic perturbation. And so to begin to look for potential mechanisms for why this might be occurring, we were fortunate enough to have RNA sequencing data from each one of these derivatives, whether it's from the patient tumor and orthotopic xenograft, which is low prime, recall, or versus the cell culture models and the subcutaneous tumors, which are more high primed. And so we essentially took the RNA sequencing data and we compared the things that were different between these groups here and then the cells, the expression signatures from the cell culture and the subcutaneous flank. And looking at these differences, we found 40 genes that are differentially expressed. So these are conserved between the patient and xenograft, but different between patient and gliomosphere, patient and subQ, as well as the xenograft and gliomosphere, or the orthotopic xenograft and gliomosphere, and orthotopic xenograft and subQ. So we found these 40 genes and we did gene ontology, and the top hits that we saw were related to these neuroglial interactions. And so that really started to get us excited based off of the work of many of all of you on the call. That don't need to remind people about all these ways in which potentially the neural signaling, whether it's via paracrine effects or through direct interactions and the formations of synapses, may influence the biology of glioma cells. So based off of this, we also searched through the literature. We also found that even synaptic signaling between neurons, a presynaptic to a postsynaptic neuron, that could strongly essentially suppress these pro-apoptotic proteins, suggesting that this synaptic signaling, even in normal physiology biology, may influence apoptotic potential. And so we started to explore this question by first harvesting neuroglial progenitors from E17 prenatal mice. And what we did is we did two different experiments. One is we wanted to understand, did they have to directly physically interact? And so we did a co-culture of the glioma spheres with the neuroglial cells, or we took the conditioned media from the neuron cultures and then added it on the glioma spheres and just looked for changes in apoptotic potential. And this is just an IF image of our glioma spheres, and then these kind of staining for these neuronal markers showing that we're able to get healthy neurons growing out in our culture. And what was kind of exciting to us is while we saw no changes in apoptotic priming between our control, so this is just free glioma spheres, and the conditioned media, when we co-culture with the neurons, we see a dramatic shift in apoptotic potential, as you can see here with this little teal color. So this is telling us that these direct physical interactions with these neurons in the neuroglial co-culture seems to be strongly influencing the apoptotic priming of these glioblastoma cells. And so we, of course, then wanted to know, is this potentially, there's a lot of different cells in this culture, it's not just neurons. So we wanted to ensure that this is actually due to the fact of neuronal signaling to these glioma cells. And so we began by adding NTTX to block action potentials. As you can see here, we see this reduced apoptotic priming when we co-culture this GBM patient-derived line with the neurons. And then what we are able to observe is that now when we come in with TTX, while it's a partial rescue, we are able to significantly rescue the apoptotic potential of these glioblastoma cells once you add in this inhibitor of action potentials. You guys and others have shown also that the synaptic signaling may be due to AMPA receptor signaling, and so we added an AMPA receptor inhibitor, MBQX, and similarly to what we saw with the TTX, the MBQX is able to significantly, although I just want to say partially, rescue the apoptotic potential of the cells when they're co-cultured with neurons. And importantly, neither MBQX or TTX has any effect on the apoptotic potential just directly on the spheres relative to control. So this seems to be an effect that is only observed, this increase in apoptotic potential is only observed when we have this co-culture with the neurons. It's also possible that gap junctions also might be contributing to this potential influence on apoptotic potential. So we added methylphenidate. While methylphenidate had a change on the cells itself without neurons, a slight change in mitochondrial priming, we're all able to see that when you add the co-culture with the neurons, that is able to be significantly higher. Again, further stressing that the point that this neural signaling that is existing between the neurons and the glioma cells in that microenvironment potentially are contributing to this apoptotic potential. And so to just summarize what we've found so far, it seems that once you are able to add in these neuron cells with these glioma cells, you're able to change the apoptotic potential from cells that are more primed for apoptosis and now are able to shift to a lower prime state, and that by essentially inhibiting the synaptic signaling, we're able to restore this apoptotic potential. And so we've been just showing this is just potential. It's not even showing whether or not this actually has some sort of therapeutic impact. It's just the potential for it to have a therapeutic impact. So we next wanted to ask that precise question, that if we co-culture our glioma cells with these neuroglial cells, does that influence cell death in response to therapy? And so we tried two different therapeutic approaches, both DNA damaging therapy with temozolomide, as well as the EGFR inhibitor. And when we look at just cell viability, when you add in the neurons, we see no significant changes in cell viability relative to the controls. With TMZ without neurons, we're able to get a significant reduction in viability. But when we add in the neurons with temozolomide, we're able to significantly restore the viable cell numbers. We see a similar effect with an EGFR inhibitor, indicating that this is not just specific to chemotherapy, that this is also potentially influencing targeted therapy responses, where we add an EGFR inhibitor, we reduce viable cell number, and then we add the neurons plus the EGFR inhibitor. We're almost able to restore viability nearly to controls, potentially indicating the importance of this in influencing the viability of cells. We of course want to do then the reverse, is can we then resensitize these cells back to reduce viability if we block synaptic signaling? And what we find is, as expected, the neurons plus the EGFR inhibitors restores the viability actually nearly or equivalent to the levels of vehicle control. But when we come in with TTX and an EGFR inhibitor or MBQX, we're able to now significantly reduce the viability relative to the neurons plus the EGFR inhibitors alone. So this would suggest that ablation of these neuroglioma synapses may sensitize these GBM cells to therapy-induced cell death. So this is all kind of this in vitro, ex vivo analysis. And so the question then is can we potentially target these neuron-glioma synapses within the brain microenvironment directly? And so we considered two therapeutic possibilities. One is by using perampanel, which is non-competitive antagonist for some of these AMPA receptor inhibitors. This is a FDA-approved anti-seizure medication, as well as meclofenamide, which we talked about is a non-selective gap junction blocker. And so to be able to evaluate whether or not disrupting these neuron-glioma synapses can influence apoptotic priming in vivo, what we did was we did ex vivo dynamic feature profiling. And so the way that this experiment works is we engrafted orthotopic PDX models. We allow these tumors to grow out. And once they've reached exponential growth, we then applied either perampanel or meclofenamide for 72 hours. We then dissociated these tumors, isolated, removed red blood cells, myelin, and mouse cells, and therefore we got a purified tumor population and did Bayesian profiling. And so again, the whole idea is to look to see how the drug treatment is perturbing the prime state in the in vivo brain microenvironment. And what was quite exciting for us to see is with both perampanel and meclofenamide treatment relative to vehicle, we see quite a significant shift in apoptotic potential with perampanel treatment. We see a similar effect with meclofenamide, indicating that these drugs are increasing the apoptotic potential of our glioblastoma cells through potentially affecting these neuron-to-glioma synapses. And to essentially support that conclusion, we engrafted a tumor in the flank and did the same experiment, this time with just perampanel. And we see relative to the orthotopic tumor where perampanel is able to increase the mitochondrial priming and apoptotic potential, we see in the subcutaneous setting that perampanel has no effect on that apoptotic potential of these tumors engrafted subcutaneously. So this seems to strongly suggest, at least from a drug standpoint, pharmacological aspect, that perampanel potentially via disrupting the neuron-to-glioma synapses in the brain is increasing the apoptotic potential of these GBM orthotopic xenografts. And so, of course, finally, and we are just kind of getting into this, is we want to understand how this is happening and really remember this apoptotic potential is just about the dynamics between these pro and anti-apoptotic proteins. And so you can influence it by shifting the presence or absence of either the pro and or anti-apoptotic proteins, and this is sufficient to kind of shift this apoptotic prime state. And although very preliminary, we just started to look at protein lysates from are parenchymal and meclofenamate treated tumors. And although it's relatively modest, and again, it's very early on, we do see that potentially these agents are disrupting the expression of some of these anti-apoptotic blocks. Namely, you can see MCL1 seeing about 30% reduction or so, and then also potentially shifting the expression of these pro-apoptotic proteins, BIM. You're seeing this both with parenchymal and meclofenamate, although again, we acknowledge that this is relatively modest effect and this is very early on. But it suggests that some of these things, that these drugs that disrupting these neuron to glioma synapses may be influencing the expression of these pro and anti-apoptotic proteins that might be responsible for shifting the apoptotic potential of these tumors. And so, again, we're a little bit early on in the mechanism, and so that's something that we really want to dig into. We've already done some global proteomics and phosphoproteomics of some of these same samples that we have treated, just kind of trying to look for pathway dynamics and see how they may potentially converge some of the neuron to glioma synapses, how they may converge on some of this apoptotic machinery that might be responsible for shifting the apoptotic potential. We want to clarify some of these studies through using some of the optogenetics. So we're collaborating a little bit with Michelle's lab to try to do some of these experiments to help kind of fortify our results. And then, of course, our ultimate goal is to identify ways that if we can target these neuron to glioma synapses, could this potentially be an avenue to sensitize GBMs in that brain microenvironment to cell death with other therapies. And so just to conclude, what we have found is that glioblastoma tumors, perhaps not surprisingly, are highly refractory to drug-induced intrinsic apoptosis. But this refractory nature, it seems to be not necessarily a full intrinsic property. By doing the functional profiling, looking at the apoptotic potential of these tumors, it seems that the brain environment uniquely drives a resistant phenotype. And this is independent of drug exposure. So we talk about drugs getting into the brain, but after we get into the brain, it seems like we also still have to deal with the fact that these tumors somehow are becoming less primed for apoptosis. And our data are pointing to the fact that maybe neuron to glioma signaling could contribute to this reduced apoptotic priming and ultimately the reduced ability for these GBM cells to undergo cell death with various therapies. And so, again, the ultimate goal here is if we are able to target these neuron to glioma interactions, we may be able to sensitize GBMs to these therapy-induced apoptosis. And we have some encouraging data that I showed today in cell culture, and the fact that we can add parenchymal and meclofenamate and be able to shift the priming in vivo also gives us hope that this approach could be at least translatable in the short term and preclinical in vivo models. But ultimately, we're hoping, because many of these agents are clinically available, that we might be able to test this in patients in the relatively near term. And so with that, I just want to acknowledge my lab. This project was entirely spearheaded by a graduate student in my lab, Elizabeth Fernandez, highlighted here. But everybody in my lab contributes to this and others' work. I want to acknowledge my collaborators, Tim Clousey in particular, but also Michelle Manji and her postdoc, Catherine Taylor, for helpful advice. This is a totally new field for us. So like it's very helpful to get feedback from experts in the field, and then to the funding and support. So with that, thank you so much for your attention. I'm happy to take any questions.
So thank you very much, David. I think that was an extremely interesting talk, and I think it highlights a very important topic, how therapeutic resistance, I think, a field that we don't know yet the role for the neuron to glioma synapses. What roles are they playing? And I think this is a very interesting framework also that you brought in here from the apoptotic point of view, how this interrelates. I think this is very interesting. I think also what we really are only at the beginning of understanding is how these different signaling, downstream signaling mechanisms of neuron-to-glioma synapses are related to the synapse. So I think that is something where we are lacking a lot of ideas or a lot of data. I think this is a very nice framework that you presented here today. And maybe I start off with a question, maybe going back again to one of the hallmarks of glioblastoma. And as you know, there are many frameworks also how to address or how heterogeneity is basically playing a role for also neuron-to-glioma synapses, but also overall in this disease. So I'm wondering how do you have any idea also, let's talking about the primed and less primed states, what are they referring to, let's say, from a single cell perspective or any other perspective? Do you have any idea or any hypothesis how these interrelates, and also how this again interrelates to the neuron-to-glioma synapses?
Yeah, that is the key question. I think that there was a question, the prompt about the heterogeneity too. We have done, and we didn't include these data today, but we have done single cell sequencing of these tumors, and we are looking at relationships between specific cellular states, for example, and at least gene expression signatures that might relate to neuron-glioma synapses, as well as the relationships to the intrinsic apoptosis. I can tell you, unfortunately, we don't have any conclusive answers for you yet, but I don't think that there's any doubt that the intertumoral heterogeneity will have an influence on this. We haven't adopted the priming assay to the single cell level. This is something that we're working on with collaborators, because I think that being able to kind of resolve that functional heterogeneity, we're really good at doing single cell exome and RNA sequencing and getting kind of these static pictures, but we really don't know what their function really is. And so I think being able to get things like priming, which is really the kind of the in-cell fate mechanism of dictating whether or not a cell will die, if we could understand that at a single cell level, both with, as it relates to transcriptional signatures, genetic signatures, but also, as you're alluding to, Varun, the interactions with the neuron-to-glioma synapses and other kind of brain-to-brain, brain-to-glioma interactions, I think this will really help us understand this process a little bit more, because as we said, there's a lot of heterogeneity between tumors and their priming potential. And so you can only speculate, and I think you would be right to, that there's going to be heterogeneity within the tumor itself. And that ultimately might lead to something growing out that you really don't want to.
Great. So, Frank?
Yeah, thanks a lot, David. That was a fantastic talk, and it's great to see how you and your team coming from, if I might say, more traditional apoptosis, cancer cell apoptosis research and treatment response, and then meet cancer neuroscience, and how well this match actually goes.
Yeah.
It's actually, so we have started thinking into this direction, because if you look into data of basic neuroscience, then you find that glutamatergic synaptic stimulation in neurons, not cancer cells, and postsynaptic neurons, and this is probably via mitochondria mechanisms, is increasing the viability of the postsynaptic neuron. So the more glutamatergic stimulation, the higher the viability, survival, etc. So, so we thought about, why not look into these kind of described mechanisms and see whether the cancer cells just reenact or exploit these, let's say, physiological mechanism of how a postsynaptic cell can get fitter. Is that something that you have also thought into, or?
We haven't messed around with kind of normal neuron signaling, but that is something that man, we'd love to kind of explore that with you, if that's something that we could help out with, because the cool thing about some of these assays, the functional assays, is you could actually dictate dependency. It's not just like, here, this is higher, this is lower, this protein, so maybe we could speculate. Like, you could actually see the dynamics of these dependencies. And so I think that would be very revealing. And then being able to relate that back to potentially, even the first question of, like, are there different subpopulations that might kind of use distinct paths to achieve this anti-apoptotic phenotype, right? So maybe a more neuronal like tumor may adopt a mechanism like you're referring to, where another tumor might use a different kind of pathway, but ultimately leading to that same endpoint of resistance to apoptosis. But again, that could have important, by understanding those differences, it could have important therapeutic implications.
I've seen you have many also IDH mutant, I think, cell lines, right?
Yeah.
So is that the same for IDH mutant too, or is that, have you looked into that?
We're just a little bit underpowered right now, but that's precisely what we want to get under. Look at is we'd love to see some of these IDH mutant, the cell lines that we have, as I think the field is familiar with, are not your traditional lower grade IDH mutants. These are transformed to a higher grade, and they often are not like, they might produce 2HG, but they don't have the features of a low grade. We do have some low grade GBM glioma models that go directly into the brain. And we, of course, can get patient samples directly. And I think that's the most powerful, is being able to analyze these patient samples. So our goal is to keep profiling these. And then you'll have all these kind of diverse molecular subsets of glioma across GBM, lower grade glioma, IDH status, et cetera, and then be able to interrogate these pathways, such as what you're talking about, and see if there are distinctions for sure.
Great. And last question. So you have, of course, shown two pharmacological approaches to, let's say, disconnect neuron-glioma connectivity and to disconnect tumor connectivity, right? This very plausible ones. Have you considered just to combine them and see what happens?
That's what we're doing right now. That's what we're doing, because you can see that there's a partial rescue with each one, and so we just don't know if that's because we're not hitting them hard enough, or maybe we got to hit them orthogonally, or different angles of it. We just don't know. That's exactly what we're doing, Frank. We're trying to figure out if that'll increase that even more by hitting kind of both sides of it.
Great. So we stay tuned. Thank you.
Thank you. Great. We have a few questions from the chat. I start with Michael Schulz's question. Maybe more philosophical questions he's asking, but do you have any guess or data which role microglia might play in establishing tumor neuronal synapses?
Such a great question, and we just haven't yet. I wish I could have a better answer for you, Michael. We haven't looked at it. Obviously, in these NSG mice, there's still considerable microglia population, and so this could certainly have a role. It's something in the brain, and so it's a hot topic that many of my colleagues are talking about, is kind of looking at the microglia and how this functions. And so hopefully we'll be able to look into that very soon, but it's a great question.
Right. Then Andreas Bigfalvi has a question. Did you do controls such as matrix stiffness and endothelial cells, how these are affecting the apoptosis of glioblastoma cells? We haven't done that. I was involved with a collaborator who made some of these hydrogels, who composed it and then changed the stiffness in distinct ways. I think we probably need to compare that directly, because that did influence proliferation. Didn't have a strong apoptotic phenotype, but no, we haven't done that specific experiment that Andreas is asking. But we certainly have access to some of these hydrogels where you can influence the tension of the ECM components and be able to see how that might influence as well. There's certainly multiple components besides these direct cell-to-cell interactions, influences of the ECM, that I've no doubt also influences the biology and the responsiveness of these tumors to drugs. Great. Then we have a next question by Arti Luchman. What stage of culture were the glioma spheres? Early cultures tend to have more apoptosis, likely mostly cell death of the differentiated tumor cells and the glia. As an additional model, would you be able to co-culture the brain tumor cells on human brain slices? Yeah, I think that's an excellent question, and it's something that we need to add. We're working on doing the brain slices. These are low passage glioma spheres because we wanted to preserve the molecular features, which we know that with later passages you'll start to get quite a bit of drift. And so you're absolutely right. We have these lower cultures, and they're maybe a little bit more sensitive at the early time points, but before adapting to the culture. So I think the questions and the suggestions that he's making are fantastic, and we'll have to check that. We're actually really looking into the brain slices right now. All right, great. So thanks again for this fantastic talk. I think there's has been a very stimulating discussion as well. Thanks everyone for joining. And yeah, looking forward to see you to the 3rd season of the Cancer Neuroscience Seminar Series. So we will announce the series soon. And thank you everybody for joining, and have a nice evening.